Hello, good day viewers. Welcome to another biology presentation. So in this presentation, we are going to answer a question that came in biology paper 2 for 2007. Okay. So the question reads, a couple who both have sickle cell trait had a child with sickle cell. Question A, 1. Explain using a fully labeled genetic diagram how sickle cell would be inherited by the child. Use the letters capital letter H and small letter h for the alleles. So this question was carrying 5 marks. So the capital letter H is representing the dominant allele and the small letter h is representing the recessive allele, the one that is carrying the disease. So here we need to understand what sickle cell is. So sickle cell anemia is just an inherited blood disorder that affects the hemoglobin. So this hemoglobin is just the protein in the red blood cells that is responsible for oxygen transportation. Okay, so how can we answer this question? So to answer this question, we need to come up with the parental phenotype. We also come up with the parental genotype. Then we can do the random crossing, okay, using the gametes. So here we are using, so first thing we come up with the parental phenotype. That is your first step. You come up with the parental phenotype. So after coming up with the parental phenotype, remember the parental phenotype in this case, we are going to say, they are saying a couple who both have the sickle cell trait. So they are not saying that this couple was sick, but they just had a sickle cell trait, meaning that these parents were carriers. So if they are carriers, it means that their phenotype is that they are normal. Okay, both parents are normal. So you say normal, okay, normal parent that will be crossed with another normal parent. According to the statement, they are just carriers. They are having the sickle cell trait. Then after coming up with the parental phenotype, we are going to come up with the parental genotype. Okay, so this is very important. That is how you are going to be given a mark. So the parental genotype, now they are saying we should use capital letter H and small letter h. So remember, these are carriers. So if they are carriers, it means that they are going to be heterozygous, different alleles, a dominant allele and a recessive allele. So this one will be capital letter H and small letter h. Again, this one will be capital letter H and small letter h. So I hope it's clear. Then from there, what else? So after the parental phenotype, we are going now to come up with the gametes. So under the gametes, we are just going to separate them. You write H here and the small letter h here. Again, capital letter H here and small letter h there. So after writing that, now we are going to have, we are going to cross them. So I hope you are following. So we will cross this with this, and we are going to have capital letter H with another capital letter H. Then from there, we will cross this with this, and you are going to have capital letter H and small letter h. Okay, then we come to this be crossed with this one. So I hope you are seeing. We're going to have capital letter H and small letter h. Then we come back. This one will be crossed with this one. Then we are going to have small letter h and small letter h. So this that you have gotten, we call this as the F1. Okay, so this will be F1 offspring. So this is the F1 offspring genotype. Okay, so make sure that you include all these stages. That's when you are going to acquire a mark. So after writing this, then we are going to have the F1 parental offspring phenotype. Sorry. So here we are now going to have the F1 offspring, F1 offspring phenotype. So this F1 offspring phenotype we are going to have. So when you look at the first child having dominant alleles, so this one is the normal. So we have normal child. Okay. Then this one is a heterozygous, the dominant one and the recessive one. So this one is also normal child who is a carrier. So this is also a normal child. Okay. So we come to this one again. This one is also a normal child. Okay. Then we come to this one, is having recessive alleles, which is homozygous recessive. So this one was a sickle child, the infected one. Okay. So this child was a sickle child. Okay. Or you can say infected. So we have answered our question. This is how we can explain fully using a genetic diagram to show that two parents who are carriers or who are just having who are carrying the trait for sickle cell, and this child was the sickle cell. Okay. Now when you look at question B, question 2 is saying what is the chance of the child being sickle celled, which is just carrying 1 mark. So here we are going to come back to our diagram right here. So look at how many children did this couple have. So 1, 2, 3, 4. So to find the chance of the child having sickle cell anemia is just 1. It's just 1 who was infected. So say 1 out of 4. So this we are finding the chance. So the chances is that 1 out of 4. 1 is the one who was sick. Then 4 is the total number of children. Then times 100%. So from here we can say 4 here 1. 4 into 100%. This will give us a 25%. 1 times 25% to give us a 25%. So this was the chance of the child who was having sickle cell anemia. So this is how you can answer this question. Thank you so much, everybody. We've come to the end of our session. This has been your presenter, Mr. Mulenga. Bye-bye.